Hey, I'm Courtney. Thank you for the introduction. Can you guys hear me? Okay, let me know if I start to talk too quiet. All right, so I'm going to start out talking about my education, background, some of the graduate research I did, because people find it pretty interesting. What I'm doing now, advice along the way, and then other routes that you can do with a similar background as what I did. And then key attributes that I think are good for scientists, and then I'll take questions or comments. And we can always go back, and if you guys have an image you want to ask questions about, or anything that I said that you don't understand or want a definition of, we can always go back to slides at the end. So I got all three of my degrees from Georgia State University in Atlanta. I actually I graduated high school in 2006 and started out at a community college where I did all my basic biology classes in a similar small class setting like this. I transferred into UGA, and that was like the big lecture halls you see on TV. Huge, easy to get lost in the numbers. I did all right. I realized that wasn't for me, so I transferred into the city at Georgia State to semi-smaller classes where I could get to know students around me better and teachers better and find a mentor to do research with. I always realized I wanted to do biology. I just wasn't exactly sure where I wanted to go because I didn't really know of any careers besides going the medical path. And you always hear about research, but I didn't really know what that meant. Oops. So as an undergrad at Georgia State, I took applied microbiology, and pretty much several days into the course I realized that that's what I wanted to go into because it's using microbiology. It makes you see it in a way everyday life is touched by an applied microbiologist. So with the teacher that I had for that course, I realized he had a research lab that I thought was really interesting. So I asked to join within the first couple days of class, and he said yes. So I started in the lab, and this lab was working on the delay of fruit ripening with the bacterium Rhodococcus. So this is a bacteria that was isolated from soil, and this particular professor had the idea of that if it could degrade compounds in soil, could it have an effect on the delay of fruit ripening? Because it's an issue how quickly fruit ripens, and a lot of it gets wasted. So when I joined the lab, that's what they were working on. They were working on if you put this bacteria in shared airspace with fruit, they saw it delays ripening. So if you see on the image right here, so this would be pretty ripe fruit, not really, you may eat it, not really. And then this was when they would put this induced bacteria in the same airspace, and they'd find they'd get several more shelf days with it. So what they also noticed is that they always would see less mold growth. You see like the mold you always see on bananas? This is a cigar tip fungus, that's what it's called. So when I came into the lab, the task that I was given was grow this bacteria, standard culture methods on plates, and then test it against tons of known fungi that grow both on plants, animals, etc. So that's what I started working on, and I found it worked against lots of plant pathogens. And so then I was tasked with to scale the process up. So the lab had a set of bioreactors. So my task was to grow Rhodococcus. So this orange thing right here. That's 2 kilograms of pure bacteria. So this is safe to be around. I'm still alive. I was around this like every day. And so what I would do is I would use a bioreactor. This is a 20-liter bioreactor, which I'll explain that more in a little bit, to grow it up so we could use it against bigger fruit trials within the lab or different applications. So one day in one of our weekly lab meetings, another graduate student asked the question, if we can kill fungus on fruit, can we kill it on a bat wing? So sort of a weird jump. So some background on that is that at that time we were seeing, and we still are, large deaths of bats in the United States. Have you guys heard of white nose syndrome in bats? No? Okay, well it gets its name. It's a fungus that grows on the muzzle of bats. You can see the image. Those are 5 bats hibernating, and they all hibernate really close together. So when one gets the fungus, it grows on its literal nose, on its muzzle. It tends to pass it around to the other bats when they hibernate in the winter, and it destroys wing tissue on the bat, and so they wake up from hibernating and they lose all their body fat during the winter. So that's the only way they survive, is if they stay fat during the winter and don't wake up. So it wakes them up, they go out looking for food, they have holes in their wings because the fungus is eating the tissue, and it ends up that a lot of bats are dying. So it started in New York in around 2005 or 2006, and they're seeing it spread south and west. And everyone at the time was really worried about what effect this was going to have on crops, because bats eat their weight in pest insects every single night, like in the summer. So they're huge. Like, there's going to be more mosquitoes if bats are dead. They're going to be eating more plants. So if they eat more plants, what are we going to do? We're going to have to spray the plants with more chemicals. So then we're going to ingest more chemicals to try to make up for all the bats being dead and not being able to take care of these insects. So as crazy of a question this was, we went forward with it. So we ordered the organism, the fungal pathogen. It's called Pseudogymnoascus destructans. This is an image of it up here. So you have mycelia, which are these. This is stain. This is really not what they look like. This is electron microscopy. And then the spores are the blue part. So we ordered it, and then the rhodococcus, the organism we're working with, this is an image of it down here. So we ordered it, and we tried it in just small cultures to see if we put induced rhodococcus, so just grown under the set of conditions that was predetermined before I got into the lab, is it going to kill the fungus in a small application like this? And we found out it did. So if you can see, this is the plate. You guys worked with plates, right? So some of you. So this is a plate with no growth on it, and then this was a plate that we put the spores of the fungus on, and they germinated and they formed these mycelia and spores. So we found out in this really small application it worked. So I continued on my graduate research to grow it in a bioreactor and make it more predictable, because the next step was to do trials on bats. So in 2015, my co-graduate student, Chris Cornelison, he started working with Bat Conservation International and U.S. Forest Services, and they started doing small trials in caves where these bats hibernated. So what they would do is they would just put the bacteria in like a cooler with several bats at a time for 72 hours, and then they would release them back into their little cages in the cave, and then they would compare that to ones that weren't given the treatment. And they found actually really good results and increased survivorship of the bats. This work is still being done. It's something you can find online. There's so much being talked about with it if you look up white nose syndrome and rhodococcus. So that's still going on, and with the process that I developed in grad school. So from all of this, what the important part was what I learned from being in grad school. It was to do something I like and that people were interested in hearing about. A lot of people did research about things that it was hard to listen to, you know. And I found that talking about what I was doing and what other people were doing with me made it a lot more fun. I learned to work with others. My work would have been a lot less interesting if there wasn't this bat trial being done with the bacteria I made. And I learned that communication is very key, because if we were doing all this and we weren't talking about it, then it really didn't matter. Because what really matters is getting the knowledge out there to people. I also learned during school that teaching is really key. Like when you're working in the lab, new students come in every semester, and you have to teach them what you know so they can help you with your project, and you guys can both continue forward on your career path. And then I learned that I was really glad that I learned fermentation in graduate school, because I accepted a job offer to start my current job 4 or 5 months before I even graduated school. So that was a huge positive, not to have to do the job hunt out of school. So right now I'm an upstream scientist at Emergent, and I do vaccine development. So this relates to what I learned in grad school, because I'm still doing large-scale productions and cultures and bioreactors. So the bioreactors I see today look more like this down here. And what I do is when projects come into us, we further develop the process to make the vaccines or drugs, and we scale them to our larger manufacturing facility, which is in Baltimore. So on a day-to-day, my day sort of looks like this. I'm just in a lab coat, glasses, and then when that process gets transferred into Baltimore, I've been lucky enough to go down there and experience it, and it looks like this. And then they're making the drug or the vaccine to actually go into humans. So it's been really neat to see that entire process. Other things I do besides fermentation are work on early vaccine platform development. So different ways to give people vaccines. And my day-to-day really looks like I come in, I check reactors, I sample, I do different cultures, I analyze data, report data, because remember, you have to communicate what you're doing, or there's really no point in doing it at all, because if you realize something's really important and you're not telling someone that matters, it doesn't really go anywhere. So some skills that I use, the benchtop skills I use, are basically all cell culture. So we do prokaryotic and eukaryotic. So we do bacteria, yeast, human cells. I've done hamster cells, monkey cells, insect cells, pretty much any kind of cell you can imagine. And what determines is when cells grow really well in culture, those are typically the ones that we use. So some of the cells that we started working with were isolated from, if you see the image on the top right, from parts of a human body, they found maybe like a cancer. So cancer is cells that grow really well, right? So scientists figured out years and years ago that these are the cells that we take and we put into culture, and we figure out the growth requirements, and then we use them to produce things for us. And typically with cell culture, you either do adherent, so like on the bottom right, you're growing things on a surface, and certain cells only grow that way. And then certain ones you do suspension, and those are the ones you use in bioreactors. Certain cell lines you infect with viruses because you're trying to produce virus, or you're using a virus to infect a cell with the molecule that you want to produce. In all of it, you have to take into account nutritional and temp requirements. Every type of cell requires different growth conditions because it was isolated from somewhere different, either from a human, an animal, or the environment. And then we do growth measurements. And have you guys done optical density in here? No? Okay. Well, basically, you measure cells in a solution by shining light through it. And the amount of light you get on the other side tells you how many cells you have. Or you count the cells under a microscope or with a machine. So the large-scale work in bioreactors, this is what a typical bioreactor looks like. It's just a large tank with a fancy name, and it stirs. And the stirrer looks a lot like a motor on the back of a boat, except it's, instead of sideways, it's just like this. And it turns. And you fill these with media, and you pump air through the bottom of it. You put probes in it that will tell you about your pH or your oxygen, dissolved oxygen, different factors like that. You have air filters to pump the air coming in and out so you're not contaminating things. And during your runs, you are always doing data acquisition. So that's the really important. You do these runs, and then you look at how did the cells grow, how much of this protein or virus did they produce, what nutritional requirements did it take, and then you use this data, and you just scale your processes up, and this is what we do. We just pick the best run of the best conditions, and then that's what goes, and we produce vaccines with that. So you also get exposure to other lab techniques doing what we do. You get to see molecular techniques, so you get to see from when it's a piece of DNA that goes into a cell, into the reactor, and then what happens to the reactor, you harvest and you purify it out with a bunch of different other machines. There's a machine for everything, so that's what makes it extremely fun. And then you get to see the analytical stuff, so that's the benchtop work that you get to look at DNA, protein, RNA, etc. Another awesome thing is all the technology that we get to see. This is it's called an Amber 250. It's automated microbioreactor system that we use. And this little arm right here is actually robotic, and it goes back and forth. You sit at the computer and you program in what you want it to do. And you have 12 bioreactors that look like this, that are about this small, all side by side. And you program them to gas a certain amount, or continue at a certain pH, or sample at this time. And it samples for you, and it'll store samples for you. So we're going like more and more automated, which has been really fun to learn all those technologies. We even have automated devices that do all of our readings for us, like pH, cell density, and different metabolites. So that's what I do, but with my background, there's so many other interesting routes I could have gone, so I'm going to tell you guys about those, and people I've met along the way that do these things. So there's basically two types of routes you can go. You can either harness microorganisms to do stuff like I do, or you can try to control them and monitor them, which will be important to prevent human illnesses. So when you harness them, a lot of people go into the food industry to produce like any of the cheese, milk, fermented foods, probiotics, enzymes. And enzymes are used as nutritional supplements for humans. They're also used in chemicals, like when you wash your clothes. There's enzymes produced by microbial systems that help to break things down better. And then lastly, then there's bioremediation. Have you guys heard of that at all? Okay, well, it's just using organisms to break down harmful things in the environment. So like when there's oil spills, microbiologists typically go in and try to figure out what organisms they could get to throw into the mix of that oil spill area or water. Could they get something to degrade that faster so it's going to spend less time in the environment, pollute humans less? So the other route, which is really interesting, is controlling and monitoring. Pretty much every product you put on your body has a microbiologist behind it, making sure that counting bacteria from samples, making sure that you're not putting pathogens all over your skin. Typically, they have an amount that's allowed, and they test every batch that goes out and say, okay, the amount of microbes in this is low enough, we can pass this, put it out to the consumer. I've met NASA's microbiologist. Never thought about it before. She was in charge of water purification on a spaceship, because it's like, think about it, you're sending only a certain amount of water up to space for how many years, and how are you going to keep that clean? So also I've met Sherwin-Williams. There's a microbiologist behind paint. Why? Because microbes degrade everything. So you want to make sure that when you're saying a paint is a certain color, that it's going to be that months later when someone puts it on their wall, and that there's not a bunch of bacteria in there degrading it. Also, the oil and petroleum industry has lots of microbiologists making sure that their product is not getting degraded as they extract it from the earth. And water quality is huge. So there's there's sources of contamination everywhere in water, and every drop of water that you've drank, there's a microbiologist measuring a certain amount of bacteria in there and saying yes, you can drink it, or no, you can't. Have you ever thought about expiration dates, like on milk or on food? That's actually based on a bacterial count. So if there's a certain amount, it's going to give it less time shelf life. And you guys are doing things like every day that you don't realize. You are a microbiologist. Everyone is. You're heating up your food a certain time to kill bacteria in it if it's been sitting around for a while. You're putting it in the refrigerator to slow that degradation down of your food. You're putting it in the freezer to slow it down even more. So, how to get started, if any of this even sounds interesting to you. You're doing it right now. You're at the top of the funnel, so you're raising your awareness of all the things you can do. And as you go through college, that sort of, it funnels, because you're going to find, okay, maybe I like microbiology, but then, okay, I like food microbiology. And then I like this aspect of food microbiology. So you guys are already on the right track. Definitely try to intern in the summers. You can look it up on government websites, college websites. Definitely go for academic research. Be brave to go ask a professor if you find out they have a lab, if you can do research with them. I don't know what I would have done if I wouldn't have asked that one professor and taken his class. I'm not sure what I would have been doing. And look for clubs and hobbies. Microbiology, you can make tons of stuff at home. You can make yogurt. You can cultivate mushrooms. You can forage for mushrooms in groups. You can do all sorts of things. And lastly, the key attributes are basically you have to be curious. Like, you have to be the person that's asking questions and not just accepting things to be true. And you're gonna have to be very tenacious and determined to do the amount of school. You don't have to do a PhD. There's tons of people that do what I do with undergraduate degrees. But if you really like learning, I highly recommend continuing your education really as far as you want to. I was in school from 2006 to 20, so 6 years for those 3 degrees. So, which wasn't bad. Some people it takes 12 years. So always know what you're getting into if you're signing up for degrees and research with someone, because it can really make or break your experience. I had an awesome lab lab boss, and I saw people that didn't, and it made it way more fun for me. Is that it? Are we out of time? That's okay. Thanks, guys.